Hello students and welcome to yet another class of mycology. In today's class we deals about the genera Pencillium, its development or the vegetative structure, the sexual as well as asexual reproductive stages. Okay, so let's get started. Now Pencillium is coming under the kingdom Fungi, division Mycota, class Ascomycota, order Uroteales, family Trichocomaceae. Now it is a common fungi which you might have seen in some of the spoiled food. For example, in the spoiled tomato, then bread, lemon, and in ear cone, etc. And it is the culture form of Pencillium. Okay, so this Pencillium is a genus of saprophytic fungi that is mainly feeding on dead and decaying materials, and they are commonly known as blue or green mold because they appear in the color of blue or green. And they are economically important because of their use in the production of cheese, organic acids and antibiotics. Besides this, Penicillium also play an important role as decomposer in the ecosystem, and they are found at various places such as soil, air, decaying food, etc. And one of the major economic importance of penicillium is the production of antibiotics. You might have heard about penicillin. It is extracted from a penicillium species. Now, some of the species of penicillium includes Penicillium notatum, Penicillium chrysogenum, Penicillium campybetri, Penicillium claviforme, etc. Moving to vegetative structure. Now, normally this penicillium, this word meaning is painter brush. It appears as a painter brush. Uh, See, it appears in the form of the painter brush. So, the word penicillium means painter brush. Okay. And the vegetative structure of Penicillium, it is a multicellular mycelium, and the mycelium is made up of highly branched, multinucleated, septate like a long thread-like filamentous structure known as hypha. So normally the fungi, you know that the body is made up of hyphae which get entangled into form the mycelium. From the mycelium, the fruit bodies, vegetative as well as sexual as well as asexual reproductive fruit bodies are formed. So here in case of penicillium, the mycelium is highly branched. Hypha is highly branched, it is multinucleate and is septate. And it appears in the form of long thread-like filamentous structure. And the cell wall is made up of glucose, polysaccharide and chitin. And the cell is multinucleate as I had already mentioned due to the presence of septa. And the cells are interconnected by means of a small coil known as septopore. Okay, the cells are multicellular. Sorry, the cells are multicellular with the presence of septa. So this multicellular cells contains nucleus and other cell organelles like endoplasmic reticulum, mitochondria, ribosomes, etc. So the vegetative structure of penicillium is multicellular mycelium, and there is presence of septa in each which makes the mycelium multicellular, and the cells are interconnected by means of small pore, and the cells are eukaryotic in nature. It contains single nucleus, then ribosomes, mitochondria and other cell organelles. And the cell wall is made up of glucose, polysaccharide and chitin. Then a brief introduction includes the asexual reproduction by means of conidiospores which are formed exogenously, and the sexual reproduction is by means of ascospores which are formed endogenously. Okay, and the food materials is mainly stored in the form of oil, globules. Now we will discuss in details about the asexual as well as sexual reproduction in the coming slides. Okay, so this is about the vegetative structure. So this structure of the pencillium look like the painter brush, and it is made up of multicellular mycelium, and this mycelium is made up of hyphae which are thread-like filamentous, highly branched, multinucleated and septate. And the cell wall is made up of glucose polysaccharide and chitin, and the food materials are stored in the form of oil globules. Now, vegetative reproduction. Not only vegetative reproduction, this penicillium reproduced by vegetative reproduction, asexual reproduction and sexual reproduction. First one is vegetative reproduction. It is by means of fragmentation. That is, at certain part, due to various reasons, the mycelium or the hyphae get broken off, and from each of the fragments develops into new complete mycelium, then into a penicillium. Okay, so the mycelium get broken off from various region, and each fragment will develop into new fungi. So this is about the vegetative reproduction by means of fragmentation. And besides vegetative reproduction, they also undergo asexual reproduction, and they are by means of conidia which are born on the conidiophore. Okay, so this is the vegetative hyphae which is septate, then it is highly branched and filamentous like structure. And from the vegetative structure, a hyphae at the time of asexual reproduction arise conidiophore. In the conidiophore, the conidium are born on a special structure known as phyalides or sterigmata, which are born on medullae. Okay, so asexual reproduction take place in the special structure called the conidiophore, and conidiophore may be branched or unbranched, and it is differentiated into medullae. This structure is differentiated into medullae, and the number of flask-shaped phyallate develop at the end of each medullae which form the conidia. So on the medullae develops the flask-shaped phyalides or sterigmata. And this sterigmata cell divide mitotically forming conidium. Okay, and this cells again divide, pushing the first conidium towards the outside, and the second conidium is formed. The process repeats, resulting in a chain of conidium found in a basipetal succession. So, from the conidiophore arise the medullae, and on the medullae, flask-shaped sterigmata or phyalides are formed, and the cells of the phyalides undergo mitotic division, resulting in the formation of chain-like conidium. And the first formed conidium are present far away from the sterigmata, and the arrangement manner is known as basipetal succession. Okay. Then the conidia may be blue, green or yellow. This conidia, which is blue, green or yellow, will impart the color of the penicillium. That is why this penicillium is otherwise known as blue, green or yellow mold. Now the conidia get detached from the parent after maturation, and they are dispersed by the wind, and they get attached to a suitable substratum, and on falling on suitable substratum they germinate by putting the germ tube, and the nucleus divides mitotically multiple times, and the nuclei will enter into the germ tube, followed by elongation and formation of septa, and it will give rise to mature branched mycelium. So in this way sexual reproduction continue. So on the vegetative hyphae, conidiophore develop. At the tip of the conidiophore, medullae will develop, and medullae will give rise to phyalides or sterigmata which are flask shaped. On the sterigmata, conidium are formed on chains in the basipetal succession, and they are blue, green or yellow in color, and at maturation the conidia are dispersed by wind, and on falling on suitable substratum, they develop germ tube. By the time the nucleus of the conidium undergo repeated mitotic division and multinucleate condition are formed and septa are formed, and it will give rise to another mycelium. So in this way, sexual reproduction continues. Next, we move into sexual reproduction. Now, sexual reproduction of Pencillium is a bit complicated when compared to the asexual reproduction. Now, some species of Pencillium are homothallic, whereas some are heterothallic. Homothallic is the condition in which male and female sex organs are present on same thallus, while heterothallic is the condition in which male and female sex organs are produced on the different thallus. So, some species are homothallic and some are heterothallic. And the male sex organ of Pencillium is called antheridium, and the female one is known as the ascagonium. So this one is the ascagonium, female sex organ. This is the female sex organ, and this one is the antheridium. The smaller one is the antheridium. Then one of the cells of the vegetative mycelium develops into ascagonium, or the female sex organ, which is unicellular, having a single nucleus. Okay, it is unicellular and have single nucleus. And this nucleus undergo repeated division, resulting in the formation of multinucleate conditions. So, in the initial stage, the vegetative mycelium, one of the cell of the vegetative mycelium, act as the ascagonium. It is unicellular, uninucleate, and the nucleus undergo repeated mitotic division, resulting in the formation of multinucleate condition. Okay. And the developing antheridium branch coil around the ascagonium, and the apical part of it is separated by septa, forming a unicellular antheridium having a single nucleus. So by the time the ascagonium develop, the antheridium also develop, and the apical cell of it is being separated by means of a septa formation. So antheridium is uninucleate, whereas ascagonium is multinucleate. And this antheridium will coiled around the ascagonium during maturation. So see, in the initial stage it is a small structure, and when it mature it is a highly coiled structure around the ascagonium. Then after maturation, antheridium bends and touches the ascagonial wall, and at the point of contact the cell wall dissolves and cytoplasm of both the cell get intermixed, and this process is known as plasmogamy. And there occurs an intermediate dikaryon phase, which is dikaryotic stage, with two nuclei present in the cell. So at the time of fertilization, the antheridium will get fully coiled around the ascagonium, and during the stage, the at the point of contact, the tip of the antheridium walls dissolve, and the nucleus from the antheridium will move into the ascagonium. So in the tip, at first the cytoplasm get of both antheridium and ascagonium get dissolved off, or they get mixed off, and the process is known as the plasmogamy. Then after that, the antheridium nuclei and the ascagonium nuclei will lie side by side. That is N plus N condition, and it is this phase is known as dikaryotic stage. So antisepta is being formed, and within each cell there will be two nucleus. Both are haploid in nature, N plus N, one of antheridium and the other of the ascagonium. So see, this is the structure discuss, the picture discuss the sexual reproduction somewhat more in detail. In the structure, this is the young ascagonium. See, it is developing. Okay, and this one is the male antheridium, male sex organ, which is antheridium. So in the initial stage, the ascogonium is uninucleate. It undergo repeated mitotic division and becomes the multinucleate condition. Okay. Then male sex organ, which is antheridium, is uninucleate. It will not undergo any mitotic division. It retain as uninucleate condition. And this antheridium will coil around the ascogonium during the development. Okay. Then this antheridium, at the time of fertilization, they comes in contact with each other. Antheridium and ascogonium will comes in contact with each other. At the point of contact, the wall will dissolve and the fusion will take place. So at first, only the cytoplasm will fuse together, and the process is known as the plasmogamy. So as soon as the wall dissolves, the nucleus will not fuse together. The nuclei, both the male nuclei and the female nuclei will lie side by side. And this process is known as dikaryotic stage. And that is 2N condition is being retained. One of the male and the other of the female nuclei. They arrange side by side, and the process is known as dikaryotic phase. And in the later part, after fertilization, that is, fertilization is the process by which male and female nuclei will undergo fusion and resulting in the formation of diploid zygote. That is, karyogamy is being taking place, where the fusion of the nucleus is taking place. Then after karyogamy, the zygote is diploid. It undergo repeated, first it undergo meiotic division, then resulting in the formation of 4 haploid nucleus. Then it undergo mitotic division, resulting in the formation of 8 haploid nucleus. And this nucleus is being surrounded by cytoplasm, and a cell wall is formed, resulting in the formation of ascospores. So during the formation of ascospores, this inside bias, the fruit body, which is the clistothecium, is being formed. So this is fruit body of Pencillium is clistothecium. Clistothecium is nothing but it is a closed type of fruit body of Ascomycota, that is ascocarp. So it is a closed type of ascocarp. Here there is no opening. It is completely closed, and it contains numerous asci, and the asci contains 8. Each asci contains 8 ascospores. So here the ascogonium, which I had already mentioned, that is, ascogonium will repeatedly divide by the formation of walls. Many binucleate cells are arranged, and the terminal dikaryotic cells swells up, forming the ascus mother cells. Karyogamy will take place, then the diploid zygotes first undergo meiosis, then mitosis. In this way, 8 nuclei are formed, and it results in the formation of 8 ascospores. Now, see, this is the structure of the matured clistothecium. It contains the thick cell wall, and inside the cell wall there are numerous asci are formed, and asci contain 8 ascospores. This is the colored picture, and it is a black and white picture. And the ascospores at the maturity, they are liberated by dissolving the wall of the ascus, and they will fall on suitable substratum, and they will develop by putting of germ tube. Now moving to life cycle, that is, which I had already mentioned, it contains both asexual as well as sexual reproduction. That is, this is the somatic hyphae. Somatic hyphae produce conidiophore. On the conidiophore, metylae are formed. Metylae will produce flask-shaped phyalidae or sterigmata, on which the conidia are produced. Conidia is the asexual reproductive spore of Pencillium, and it germinates by putting of the germ tube, and it will give rise to somatic hyphae. This is the asexual reproductive cycle. During sexual reproductive cycle, the male sex organ antheridium and the female sex organ ascogonium are formed, and the antheridium is uninucleate, whereas ascagonium is multinucleate. The antheridial branch will coil around the ascagonium at maturity, and the fusion resulting in the formation of diploid zygote. Okay, at first the plasmogamy will take place, then the karyogamy will take place, or the meiosis will take place. Fusion will take place, and the diploid zygote undergo meiosis, then undergo mitosis. Numerous ascospores are formed, which are arranged in the asci, and this asci are produced in the fruit body known as the cleistothecium. The wall of the cleistothecium is known as peridium, and it is a closed structure. Cleistothecium is a closed structure containing asci which contain 8 ascospores. And at the time of maturity, the peridium will broken off, asci wall will dissolve, and ascospores are liberated, and on falling on suitable substratum, the ascospores will germinate by putting the germ tube, and this will give rise to somatic hyphae. So in this way, the life cycle of the penicillium will continue. Okay, so this is about the penicillium, its vegetative structure, asexual reproduction and sexual reproduction. Thank you very much.